Ma'am, you can start exactly at 1:30. Automatically, when you share, the video will be stopped. Okay. We have just 1 more minute.
So I can share my screen after that.
Yeah, yeah.
Okay.
Yes, ma'am, you can.
Hello everyone. Welcome to LLB International Career Talk webinar series. Maybe we'll wait a minute and then start.
Yeah, yeah. Yeah, you can start.
Yeah. So the slides are not visible right now. Yeah, now it's visible. So, okay, good evening, good afternoon, everyone. Welcome to the LLB International Career Talk webinar series. Today we have Dr. Meeta Sunil with us, and she will be talking about her career journey and share her knowledge about working in the area of genomics and how is it as a career choice. Dr. Meeta did her master's in botany from Delhi University, followed by PG diploma in bioinformatics from Institute of Bioinformatics and Biotechnology. She also
Sorry, Snehal. Somebody.
yeah. Can someone's audio is on, microphone is
Yeah,
on. Can you please mute it?
yeah. I
Even I should mute
I think
mine.
it's from the Meeta ma'am side, I guess.
Should I mute myself right away?
Yeah. Yes, ma'am. I can do that. But when you speak, you can unmute. Okay, ma'am. Yeah, yeah, yeah. Snehal, you can start.
Okay. So I will start with Dr. Meeta's introduction again. Maybe some of you might have missed it. So Dr. Meeta did her master's in botany from Delhi University. This was followed by PG diploma in bioinformatics from Institute of Bioinformatics and Biotechnology, that is IBAB. She also did her PhD from IBAB with focus on sequencing and annotation of Rajgiri genome. After this, she joined MEDGENOME Bangalore, and she is currently working on designing, developing, testing, and deploying NGS assays for challenging problems. As I understand, she has worked on both wet lab and dry lab aspects of genomics, and especially currently in MedGenome, she is involved in studying genetic variants in patient data. So before going further, we have a few things to share with you, a few announcements. So Dr. Meeta, can you go to the next slide? Yes. So we are LLB School, and in Futura, our vision is to connect students, active researchers, and professionals belonging to the fields of biology, bioinformatics, and its allied subjects globally. We provide every possible service to bring the biologists and bioinformaticians together under one single roof. Uh, and our website is llbschool.org, which you can go to and get yourself registered as collaborators, freelancers, tutors, or expert consultants. Uh, the more there are, the more we can offer to each other. And on the technical side, we have a request that please kindly turn off your webcam or mute yourself for better webinar session. and try to note down your questions, which will be addressed at the end of the session. And you can post your questions in the chat. And please subscribe to our YouTube channel and confirm your presence. This time we have a bit of difference in the feedback forms. So there will be a quiz regarding the webinar that is conducted today at the end. And those who score more than 50% in this quiz will get their certificates. And the feedback link for the quiz is already shared with you on our Telegram channel. And kindly note that we will not be responsible for any typos on the certificate. So please be sure to spell your name without any mistakes. Please go ahead, Meeta. Yeah, and we would also like to thank our collaborators, Center for Plant Molecular Biology and Biotechnology, that is CPMB, TNAU, and IMOT AgriForum. So we are all really excited to hear more about Dr. Meeta's journey and her insights regarding genomics as a career choice. So I request Dr. Meeta to please go ahead and share your talk with us.
So thank you, Sneha. And I would also like to thank LLB and Mahesh for organizing this and giving me this opportunity to talk about my own little bit of experience in this field. As I know from the colleagues here in LLB that this small little platform has been developed for providing a general career guidance to students from various backgrounds and at different levels of their education and experience in their profession. So I believe there are students from 12th standard students, undergrads, postgrads, and even higher up. So keeping that in mind, I have tried to put my little 2 cents in a few points. First, I would like to tell as to how do I think is the overall career perspective inside for entering into the genomics field with respect to your qualifications. And then I would like to talk a little bit about what my education qualifications and professional journey has been. And then a little bit about what is the basic skill set that is required to be able to thrive in this field and also excel. And then a little bit of why do you actually want to be in this field and what are the different opportunities that we can, as of now, which are available. And then we will be open to all Q&As. So I've tried to put this in a cartoon. So the title of the talk is climbing up the DNA ladder. Here I mean to, I'm trying to say DNA ladder for genomics because the genome is where the DNA is. The study of DNA is called the genomics. And climbing up the DNA ladder as to trying to see your career into the field of genomics, starting it and growing into it. So this is here. I would like to start with a very basic education in the field of science is what is essential. Thereafter, you can grow your skill set as much as you want, and you can enter the field of genomics at any stage. So first, nowadays, we have bachelor students also getting hired up in companies. So mostly when I say here bachelor students, they can be directly hired as interns or trainees or bioinformatics analysts in companies, or they can be they can go ahead to pursue diploma or masters and then get into corporate world, or they can get into still research mode, still a higher level, get into a research mode as junior research fellow or senior research fellow. After that, also they have a choice to go to either corporate or to the academia. So within academia, they can either do their postdoc. They can either go to continue in academia with a doctorate, with a PhD, or can get into a corporate sector and work as even without PhD, you can work as a programmer or into operations. We can talk about this in a little detail. And after after your research fellowship, you can, after your PhD, you can either again go ahead for a even higher research field, as in for a postdoctorate, and then eventually go into a corporate sector again, or get into universities or government institutions. So in India, we do. I'm focusing my context here in the sense of India. What opportunities are there in India specifically? So you can either have you can either get into teaching or research within universities as faculty and professor eventually, or you can get into government institutions like DRDO or other CSIR institutions and get into a research scientist mode. And other than that, within the corporate field, usually in India, the corporate companies that are there, I have a list of them at the end of the slides also. So there in India, we have very few companies which do a lot of research work. So most of the work is concentrated into operations. So operations basically means that you have a set of defined methodologies for analysis of the data on which that company works. So that data could be from plant genomes, or it could be from microbial genomes, or it could be from animal genomes, or it could be from human genome, depending on what the primary product for the company is, whether it's a service provider or a product developer. So you can have BTech students, MTech students, MSc students, diploma students directly hired as analysts, bioinformatics analysts for the operations teams, or for programmers, because you do need help of scripting or programming languages to solve your bioinformatic problems. Or if you have a lot of work experience or doctorate or postdoctorate, then you can directly get into the research mode for the companies. So this is like an overall scenario which there are different possibilities with which you can enter into a company or into the academia with the point of view of growing into the field of genomics. So talking about my own journey, it begins with my basic educational profile, which starts with the higher education into high school into science stream with the biology in 10th and 12th standards, and then my graduation, post-graduation in botany. And thereafter my post-graduation, I was looking for I was not clear in my head at that time as to what I wanted to do. So I was exploring. So I explored the field of intellectual property rights. I have a couple of certification courses from WIPO and a distance education diploma program from NLSI. And it is also a very, very interesting field and related to everything that you do. Every field that you enter into is associated to IP in some way. So this is also helpful. Is there a question?
No, Dr. Meeta, you can go ahead. We will take questions at the end.
Okay. So IP rights, they are also important and associated, especially in the field of biotechnology also. And then I had a opportunity to get into a professional course in bioinformatics in the Institute of Bioinformatics and Applied Biotechnology in Bangalore, which was like a turning point in my career, where I was exposed to a lot of career opportunities and different domains within bioinformatics, which are really interesting and challenging. So after my PG diploma, I got a project from my PhD guide. She was working on sequencing of Amaranthus genome. So looking at my background and skill set, she offered me the PhD position, and I was fortunate enough to have accepted it and worked on it. So that is about the educational profile. But the real steps into the field of genomics started only in the PhD phase. So my PhD project was sequencing and annotation of Amaranthus hypochondriacus or Rajgera genome and transcriptome. So some people know it as Chalai in Hindi or Rajgera or Dhante Soppo in Kannada, Mulaikira in Tamil. So if you're familiar with these words, you will know this is a very regular plant that we eat at home. But the seeds of this plant is very, very rich in an essential amino acid called lysine. And the reason behind that is not known. So genomics is such an interesting and efficient tool which helps you, zoom into the DNA and find out possible reasons of the possible phenotypes that you happen to see. So our aim was to dig into the genome of this particular plant and try to understand why did this plant have a lot of unique traits and especially high lysine content. So that was a 5-year experience that I had in the PhD program, and I was able to work on building a. So just before I get into the details of what I did, this whole project was based on a technology called next generation sequencing. This is for the younger people who may not know about NGS. So there, the entire DNA of a species is broken into smaller fragments, and every fragment is sequenced, bit by bit into a sequencing machine, and that data is converted into a digital data. The organic data from test tube is converted to digital data in the computers, and we get to analyze them using different tools and methodologies. And there is where programming also comes very handy. So I was able to generate draft and near reference genome for the species along with a transcriptome. And we worked on different technologies like Illumina and PacBio. And then we did a lot of annotation for all the sequenced fragments that we got for the plant. And we did a lot of comparative genomics also along with other plant species to tell that the species that we sequenced was correctly sequenced and analyzed, and that what are the features in this plant compared to the genomics of other plants. So how does it stand out? For that, there were different methodologies used, and how was the evolution happening within that plant in comparison to other plants. So characteristics of that genome, this whole genome duplication, heterozygosity are certain characteristics which were deduced for that specific genome, and phylogenetic analysis was done for comparing the evolutionary side of this plant with other species. And again, you get to see gene expression profiling. So when you study the RNA, you are able to find out what specific genes are expressed to what level in a given condition. So this also is a very important and efficient tool to give you an idea of how an organism is behaving at a given time point. And there are certain structural reorganizations also observed in different genomes. So there were also some transcript fusions which were identified in this plant, which also gave it a certain edge over other plants. And then we also did a lot of other work, which included development of an in-house tool for predicting the proteome based on other homologous sequences. And I was also fortunate enough to have gained experience not only in computational work during my PhD, but also in the lab assays and experimental work. So we did all the extraction, quantification, and gel visualization of the nucleic acids, estimation of proteins and amino acids by HPLC, which is a high pressurized liquid chromatography technique, which is a very high-end technique and a good experience to learn. And then library preparation for sequencing and doing the sequencing, things like that. So these are some of the main highlights that I have written. And as and when you keep working, you tend to expand your skill set and accumulate more, which helps you eventually. So this was a very good experience. And along with that, I also had experience in doing farming and collecting the tissues and storing them and preparing a herbarium, which is also a very important part, which makes you learn as to whatever you are working on, you have to save that so that people can validate your work and regenerate the results elsewhere using the samples that you collect. And it is very important part, but a subtle part. And then I was also, during the PhD, you also get an option to interact with the junior people and your junior students in your institute and help them learn a little bit of what you know and help them with their projects and all. So that was an overall PhD experience, and it also helps growing further in the career, whether you go into academia or into the corporate sector. So soon after my PhD, I was also fortunate to get a job into MEDGENOME directly. So I was hired as Associate Bioinformatics Scientist too. And here there was a complete shift of the application of genomics in my professional path. So during my PhD, my application was focused on nutritional value, understanding the nutritional value of amaranthus with the help of genomics. And now the focus in MEDGENOME shifted to human genomics. So understanding how and understanding a particular disease type, the mutations behind it, and how to diagnose that, all those things were a complete shift of focus and complete shift of application of genomics. But the basic techniques and understanding and concepts and methodologies remain same. So in my opinion, if you understand the basic techniques and the technology per se, you can apply it to any specific field, whether it is microbial genomics or agri-genomics or human genomics or animal genomics. So there are varied applications. So some of the projects I got to work on were understanding certain disease types like muscular dystrophy, specific eye diseases, a very rare and fatal autoimmune disorder. And then there are other types of projects where you need to benchmark different types of tools for calling different types of mutations. And our strength over there was identifying certain structural variants from whole exome data, which is very challenging. So owing to its own the type of rearrangement that happens in the DNA, based on that, the method of identifying that mutation is different, because now the DNA is not like a single thread, it is in bits and pieces and millions of copies of that. So you have to, it's like a needle in the haystack kind of a thing. So you have to look at different methodologies to confidently tell that a particular type of rearrangement is present or not. And these types of rearrangements are very important in certain types of diseases. So, and especially in clinical diagnostics, it is very important to identify some characteristic mutations or novel mutations. And then there were other projects where we had to benchmark and study different technologies for variant calling. So you get to keep learning newer technologies and get hands-on on those. And of course, because we were in the research team, we had to write papers and also mentor the new joinees there. So for some time, this was the kind of experience I had. And then eventually I was upgraded to Bioinformatics Scientist 1 and more responsibilities came in. So more types of applications came in and more time and more in-depth understanding of things came in. So the types of projects again that I started working on was designing, developing, testing, and deploying production quality methodologies for NGS for diagnosis of specific mutations. So so this was something newer than what I was doing before. I was identifying specific variants from a family data or from a single person based on already established methodologies. And now I was developing newer methodologies. So these are the different types of work that you get to do in a company. And MEDGENOME, being a diagnostic company as well as a research company, has both aspects to it. So you can work into operations on well-defined methodologies for regular analysis of patient data, or you can work into the research team for developing newer methodologies or newer products. Or there is also a commercial team which is like a service provider for analyzing data for academic institutions. For example, if somebody's PhD data is there and they need help for analysis, all those things also happen. So the newer other tasks that were happening, since MEDGENOME is also a product developer, we were developing a lot of new exome panels and other lab assays, which needed to be validated from time to time. So that is also another thing that we need to see. If we are coming up with a new methodology or with a new product or a panel or an array chip, we need to see that we need to see the QC first for quality check and quality control, and then see that the results from this are reproducible, whether one person does it at different time points, or two different people do it, or they do it on two different machines, or they do it on the same sequencing machine in two different lanes, things like that. So that is called validation, where you, and also the results here have to match with the experimentally established values which are already there. So this will become like an end-to-end validation of all your products or new assays that the company launches in the market to the clinicians for testing. And then my work domain involved not only germline Mendelian disorders, but also somatic disorders, most of which are cancer samples. So all oncologies are somatic disorders, and all Mendelian disorders are germline. So we built an in-house database for annotation of somatic variants, which was not available as such. So so whenever we do a variant, whenever we try to do analysis for a given type of germline or somatic sample, the variants need to be annotated, as in certain level of information has to be associated with all the variants identified in that sample. By sample, I mean an individual or a patient. And for every variant, when we annotate them, it helps the clinician take a call whether that variant is good enough to cause that phenotype, the disease phenotype or not. And if it is a critical variant, what type of treatment plan should be done for that patient, whether the patient will respond to a certain drug or not, whether he will or she will respond to a therapy or not. All these decisions are made based on those types of specific variants which are identified. Then there is another project where you have blood typing. So blood typing is a field again, like we know we are either A positive, B positive, A negative, B negative, or AB or O blood type. So these are the most commonly known blood types. But other than these, there are 40 to 50 more blood types for a human body. And so blood typing plays a very crucial role when there are patients who require blood transfusion on a regular basis or an organ transplant. There is also HLA typing for that. So these typing methodologies are very important, so that these are specific markers for that individual, whether that individual will be able to accept the blood from another individual or not. So such types of projects also we do. We have to develop a methodology for doing blood typing. And then regular for all the cancer-related samples and projects and analysis methods that we do, we have to address the day-to-day challenges and clinical reporting of those, and also come up with newer products in those areas. So this is what basically I'm doing these days in MEDGENOME, and it is a very great experience and a lot of learning experience too. So other than my own professional journey, I would also like to emphasize on the basic skill set that any student would need to start a career into genomics. And like genomics sounds to be a very high-end domain, but it's a very simple tool, and anyone can do it provided you have an interest in that. So I would focus on three main categories. In fact, the first category is the most important one. If you have those, you can do anything in the other two categories also. So first and foremost is you should have an interest. If you understand the field, why is the field important, and if you get intrigued by genes and DNA and proteins and things like that, then that interest is something that can drive you to do well. And there is no formal education required other than basic knowledge of science for genomics. So you should be able to do self-learning. If you're good at Googling, you should be able to Google out and read a lot of published papers, articles, and self-learn about certain newer topics, because there are too many newer things to learn in genomics, and rather for that matter in any field. And with time, these technologies also grow very fast and newer technologies keep coming in. So you have to keep yourself updated. So you need to be self-learner. You need to be able to implement those things a little quickly. And you should be able to troubleshoot for impasses if there is any um, problem in your script or while running anything, or if you have an impasse in your analysis, you're not getting the desired results, then you should be critically thinking of other possibilities of how you can analyze, or what is a different method that you can use instead of the conventional one. So that way, keeping yourself, your mind lit up will help. And of course, patience is a key, because this is biology. Within biology, there is nothing perfect. So you will get a lot of impasses. But every situation is very challenging as well as very, very interesting. So if you're able to, you know, do this self-learning and keep your interest in, and then have a little patience if something is not working out, look at other options. And there are a lot of ways you can think of when you start working, when you start having the hands-on, you will have a lot of ideas as to what all you can implement to get your task done. So these things are important, and then cross communicating within the teams or beyond, us talking to people who know about certain things, presenting your work to other people, keeping in pace with technology and enhancing your own skill sets with time, doing managing your projects, using some management tools also nowadays. So these things are basic things that you need to get going for an everyday, professional life. And excuse me. For your computational skills, I think only there are two basic things that you should be familiar with, if not very well versed. You should know a little bit of Linux commands so that you can, handle all your digital data on a daily basis. Do a little bit of look at the data and understand what it is. A little bit of manipulation, as in parsing of the data to interpret what we want to. So for that, some handy commands are good to know in Linux. And one programming or scripting language you should be comfortable in. For example, I started off with Perl, and then eventually Shell and Perl are my strong points. And now with the time, I'm also starting to learn Python. Some people are very good in Java. So any one language is good for bioinformatics. Mostly Python is taking up now. So and Python is very easy and user-friendly programming language. So if these 2 points are satisfied, you are ready to start working right away. And of course, a basic education in science stream. So in my experience, I have seen there is no specific subject or it's not necessary that you have to have a background in bioinformatics alone. I have seen people from varied backgrounds entering genomics, even in IBAB for the PGTB or master's program. There were people from all different backgrounds, from veterinary science to MBBS, veterinary science, botany, zoology, chemistry, all different backgrounds. And I have seen people from physics and chemistry and mathematics and computer science getting into genomics, and people from biology also going into IT fields. All these things happen. So genomics is like a place where anyone who has an interest and can have a little bit of hands-on with Linux and one programming language and an interest in biology can just start working into that. And as and when you get experience and you want to grow in the industry, you will definitely have to advance your skill sets. So you can, optionally have develop an understanding of bioinformatics and sequence analysis and databases involved. The NGS tool, which is there, the next generation sequencing methodology, the data that comes out of it, how does it look like, what is the QC to be done, how is visualization happening, what do you understand from that, a little bit of statistical tests to give significance to your result interpretations. Nowadays, data science is also picking up. Data science is something which is involved in every computational field, not just genomics. But because genomics also deals with a high amount of data, there is a direct implementation of data science within genomics also. So this is also a new area which is getting integrated into genomics, and there are a lot of lot of good work happening here already, and there is a lot more scope further. And next flow is something I would like to introduce you all if somebody is not familiar with it. So this will Nextflow is a specially designed software suite, you can say, for bioinformaticians, especially for genomics field. It helps you manage your operations very well every day, launching of jobs and managing them. If something breaks, then redoing them, visualizing how the processes are going on. So technical part of Nextflow is very good to help with that one. And of course, because you're dealing with high throughput data, distributed computing is also very integral in this field. So, excuse me. So you have to work with HPC clusters, whether it's from SandGrid Engine or PBS or Hadoop. So there are specific ways you need to modify your codes to make them compatible with this. And it helps you distribute your jobs, which are very memory intensive or time intensive or CPU intensive jobs. They get distributed onto different computer systems, and then your runtime reduces many of those. Like it may go down to 1/10 of the total runtime or lesser than that. So these things are some things which are picking up and are very handy if you want to grow further and quickly into this field, which I think is required these days. But these are not essentials. These are advanced skills that you will automatically gain when you start working into this field. And this all skill set for computational side is only for people who want to go into computational genomics. And for people who want to get into the experimental side of the genomics, they need to be well versed with the molecular biology assays in the lab and their handling and management and regular QC and everything. So these are all the basic skill sets which are required. So in addition, I would just like to reiterate the major fields where genomics is playing a crucial role these days that we really get to see all the time around us. The first and foremost comes as personalized medicine. So many a times we get to read news articles or even in published papers, some cases where there is a very, very rare genetic disorder that a person has and there is no medication or treatment available to that person. So for those cases, they quickly do sequencing of this person and try to find out if there is one or two or even more significant variants that are contributing to that disease. So once the variant is identified, it becomes a lot more easier for the doctor to take an informed decision for the treatment and prognosis of that patient. So personalized medicine has become a very, very robust tool as an application of genomics for treating rare disorders. And so there are the clinical diagnostics, oncogenomics, all these are certain subsets of personalized medicine. So clinical diagnostics is what I am involved in right now. So this is mostly catering to rare genetic disorders again. In diagnostics, we usually develop newer products for diagnosis of different types of diseases. These products are in the form of new panels, arrays, chips, things like that, or methodologies. And in oncogenomics, we try to apply the concepts we try to utilize the methods of genomics for treatment of. Here, it's not about diagnosis. It's more about alleviating the condition and handling the cancer relapse cases. There are specific DNA markers that are involved that are related with the relapse condition, or that give a direct idea of what kind of drugs will suit that cancer patient. There is also a lot of effort going on in the field of oncoimmunotherapy, where the patient's immune system itself is being utilized to target the tumorigenic cells, the tumor cells. So with the help of genomic tools, there's a lot of work going on here, and it's very critical. And another thing is another application within personalized genomic is also you can identify the risk profile for an individual for a given disease. For example, how likely is a person to develop CAD, coronary artery disorder, so or any other disease, Parkinson's or anything. So there are certain statistical measures which are calculated for that individual. So they create a cumulative score based on, again, different markers within that individual and in comparison to the common variants seen in populations. And based on the score, a certain level of risk percentage is associated with that individual. So that is about developing a risk profile. And for all of these studies, another thing that companies work on is developing databases or developing ethnicity-specific reference data sets, as in population-specific genomic markers, the set of genomic markers for a specific population. So if we are studying an Indian cohort, we cannot just utilize the publicly available Caucasian cohort. So we need to know what are the normal mutations that happen in individuals within India so that we can rule them out and pinpoint to the specific disease-causing variants in the Indian population specifically. Or within India, it could be a Gujarati population versus a South Indian population. So ethnicity also plays an important role. And consanguinity, these things play a very important role. So carrier screening and prenatal testing is another application. So when I mention consanguinity, if there is marriage between first cousins or related people, they will be more likely to carry certain disease markers in a dominant expression than other non-related, non-blood related individuals getting married. So carrier screening is also one of the applications where there may be a history of miscarriages because of genetic reasons. So the parents are advised, especially the mother is advised, to undergo genetic screening to avoid a miscarriage in the next conception. And prenatal testing also similarly, whenever in the 3rd month of pregnancy, there are a lot of tests that happen to check the genomic condition of the developing baby. So prenatal testing is also part of that one. And HLA typing, blood typing is also what we have already mentioned. It's very important to characterize whether two individuals who will be involved in organ donation or blood transfusion, whether they are whether they match or not. Otherwise, if the HLA types do not match, the organ transplant will fail and it will be fatal for both the people. Similarly, for blood transfusion, if the blood markers do not match, if the blood groups, there are 50 plus blood groups, which if any of those do not match, then it can be critical for the patient's health. Then nutrigenomics is again another thing which we have discussed before. This was where I was doing my PhD also. The main aim is here improvement of the crop species, either with the any specific trait of that plant species needs to be, which is agronomically or industrially important, that needs to be improved. Sorry. So then genomics also helps in understanding the phenotype, the genotype behind those phenotypes, and manipulating them to get the desired results in terms of genetic engineering of the crop plants. Then again, similarly, genomics of medicinal plants is also helpful because it helps us understand the medicinal plants better and utilize them better for human use. And similarly, phylogenomics is another domain within genomics where evolutionary studies are made based on the sequence analysis of many different species taken together. So that also helps us to understand how the evolution happened and where in the evolutionary time and space does the specific species fall, and how we can benefit from that species, what will be the related phenotypes for that species. So that is another field that helps us understand where we can apply genomics and gain some useful insights. Then understanding a single genome nowadays, everything which we see around us is getting sequenced from cockroach to different types of plants. Sorry. And amaranth is what we worked on in my PhD. Cobra genome has been published by MEDGENOME in association with AgriGenome. AgriGenome is also working on tiger genome, but it's very difficult to get tiger tissue as such. But this is a wild tiger genome that they want to do based on the fecal remains of the tiger and not the tiger DNA and the tiger tissue itself directly. So these are different types of applications. I have forgotten to mention metagenomics here, but I'm writing here understanding the infectious agents. So this is in the context of human diseases, again, based on bacteria and other viruses. For example, COVID-19 right now, a lot of genomic work has been done and a lot of genomic data has been developed for studying this COVID-2 viruses. And there is a lot of work going on in MEDGENOME and other places across the globe on mycobacterium tuberculosis. So main challenges with microbial assays are most of them take very long time for getting the test results, especially MTB takes months altogether, 6 to 9 months for getting the test results, the lab test results. So genomics, again, is a very handy tool here because within a week's time, you can diagnose whether the MTB is active or not, whether the person is infected with MTB or not, and what type of a strain is it, and what medicines, medicines are it resistant to. So which medicine will suit the patient, which will not suit. All these informed decisions can be taken based on the genomic profile of these microbes. Then metagenomics is again another field where for example, gut metagenome or air metagenome, all these things will help us understand what is the combination of microbial load, what is the combination and load of microbiome in that particular environment. If it's gut, then in the gut. If it is air, then in the air. If it's a water sample, then in the water sample. And how does it affect our health, and what are the different characteristics that we can find out from that one. So these are some of the most dominant applications that we are seeing around us in companies and academia. Other than that, we can also think of a lot more. So I've just listed down a few of them here. Proteomics, metabolomics is also one field where they are linking genomics to the protein and metabolic substances. So not just the results from genome data. The results from genome data are also matched with the results from expressed proteins and the metabolic pathways which are involved, and what all other metabolic agents are involved in the pathway. So all those data together are analyzed, and they add more value to the genomic studies as such. So this is a more bigger version beyond the genomics, where other fields also come in collaboration and give us more promising results. So these are some of the main applications which I had listed, just to show what are the different possibilities if you go into academia or companies. What can you expect? What different types of work profile can you expect for that, and how important it is? And then I would like to end my talk with just a little bit of, the places where you can look for working into these different types of applications. So within the company side, these are a few of the companies which are listed, but there are many more startups coming up. So MEDGENOME, this is Imperial Life Sciences, Inference, Flavors Life Sciences, Thermo Fisher, Syngene, Perkin Elmer, Elanco, Strand Life Sciences, DuPont, Equia, Monsanto. These are different companies across different places in India. And then here I have one government initiative by DBT. And these are all the government institutions which are taking part in this collaborative effort. This is again showing ethnicity-oriented task, where it is the aim is to develop a catalog of genetic variations specifically in the Indians, so that it is useful in treating, in understanding the Indian patient data better. So there are a lot of CSIR institutions and University of Hyderabad and CCMB, many other institutions which are part of this. And these are the places where you can apply for an internship or training or a job or a research fellowship based on your skill set. So with this, I would like to end my presentation and open up for questions.
Thank you, Dr. Meeta. So we already have few questions here, and I will start with Akhilandeshwari's question because I think you have already answered it here. So her question was, I have completed PG in bioinformatics and I want to enter into genomics, but I don't know where I can. Could you please guide me, and can you please suggest the bioinformatics companies for genomics? So as you can see, we already have it on our screen. So I hope Akhilandeshwari, you can take note of these and try to look into the jobs that are already advertised at their websites and see if it suits to what you want to do with your career going ahead. Do you have something to add, Meeta?
I think I can just add a little bit here that
Yeah.
these are all the companies you can target, but you can also create your LinkedIn profile and be open to recruiters, and also update your CV with your skill sets and put them on the job portals, because the companies also majorly look through job portals only, so you will get a lot of response from there.
Yeah, that's right. So then more on the general aspect of genomics, we have a question from Annapurna, and she asks, with human genome validation, will it be possible in the future to cure genetic disorders like Down syndrome? What do you think about it? Is it possible to cure them?
Down syndrome is because of a complete chromosomal abnormality, not a segment, a small segment. So nowadays, newer technology of gene editing are coming into application. So, but they target a very small region of the genome. The whole chromosome, as such, readily we cannot think of. But eventually, with more advancements in the technology, this is something that may or may not happen, but it's a very complicated thing to address a whole chromosome. But with genomics, as such, computational genomics, it may not be possible. But with experimental genomics, it may be targeted.
So, but maybe not for Down syndrome, but for
Yeah.
other hereditary diseases. Already, I think the research is underway at
Yes, yes.
various institutions.
Yes,
And
CRISPR-Cas is being utilized already, and it is just in the nascent stage. But we do hope to see a lot of success here.
so then we always get these questions, and here again we have this. So the question is, how can I enter into bioinformatics field after completing either my BSc in biotechnology or BTech in biotechnology? What do you think I should do to say shift from a biotechnology degree to get into bioinformatics or genomics career?
So, if you directly jump from graduate level to companies or academias, you will be offered the position of trainees and interns only. And eventually, after that experience, you can either directly go and do a internship with a company or with a university, or you can go ahead and complete your higher education at least till the master's level, and then jump into industry or academia, which will give you better scope. But at the level of graduation, it is a little rare that you get a proper full-fledged job in life sciences field directly, but it will definitely help you to get internship, get work experience in the form of internship. So if you want to get into the field, like hands-on directly, then internship is what I would recommend.
So, and particularly, I think for biotechnology students who may not have as much experience working with, say, computational tools, I think internships are a must before they can, I think, start
Yes,
working.
because you need to have some hands-on.
Yes.
Otherwise, companies won't be able to trust that the resource will be able to contribute thing.
That's right. And yes, another very recurrent question: uh, do we need PhD degree to get good package or maybe growth in genomics field?
PhD is not mandatory. PhD is something optional. It depends
Yeah.
on you want to see your career profile. People who are doing MSc or mostly MTech actually, because companies tend to, there is a kind of a mindset that MTech people will be more equipped with programming languages and all. So, so MTech students also get a good money these days in all of these companies which I've listed. And I think you shouldn't be deciding this based on the package that is being offered because package will be fair
Mhm.
based on skill set. So you can either do directly a job after masters and your work ex counts. So if you do 3 or 4 years of work ex, if you have, it will be equivalent, not completely equivalent, but within the corporate sector, it will be equivalent to a PhD experience because the work experience matters a lot. And if you do a PhD, you will get placed at a higher package compared to the number of years of experience that way. So, but package with respect to your skill set and degree is always proportional. So, I think both options are good. Either you do masters and jump into the field, or you do PhD and jump into the field, or you do a masters, get 2 3 years of work ex, and then go to PhD and jump back into the corporate sector. So all possibilities are good.
Yeah, so I think the kind of skill sets that you can offer to your employer, I and your experience, that is what matters
Yeah,
than
yeah,
just
yeah.
having a degree. I
For
think that's
example, in MedGenome, we last year we hired one person who is only a tech, but he was very smart, had a little familiarity with data science, was good in Python, so he can contribute. He didn't have any knowledge of NGS, and he was only a BTech student, but he was also hired at a very good package for a beginner.
yeah. So that is a good point to note that students can enter into genomics at various stages of their career, either after bachelor's or master's or after PhD. But yes, what counts is their experience and the skill set that they have acquired through over the years. And yes, so then going to the next question, just let me check what all questions we have. I think we have covered most of them. Uh, there is one question, whether do you know about any government jobs that or academic jobs that a person with genomics background can like, get?
There are a couple of institutions. All CSIR labs also offer government positions like scientist A, B, C, D. These are the positions that are there. But there are a couple of government institutions which offer this, and they keep coming on LinkedIn also from time to time and at various job portal sites. Even in universities, you can get a scientist position. You can, and NIMR is an institute, National Institute of Malaria Research. There are institutions, but I have seen that the government institutions have just started working into genomics from a few years lately. But corporate sector, you have much more opportunity available right now. Uh, but government sector has also taken up, and there are options available.
There is another question asking whether do you know any institutes or colleges that provide, say, direct placement after, say, graduation or masters related to genomics or bioinformatics, if you are aware of any such institutes.
I think nowadays almost all the institutes are doing this. My alma mater IB is definitely doing it. But other than that, other than that, all the when we hire students in our company, they are from triple ITs, IITs, and other institutions also. So all these institutes do campus placements only. And when the campus placement happening, the package and all is also good because the campus takes care of that.
Um, then there is another question. How do you think the field of epigenomics is playing out in the field of cancer in India?
Yeah, I missed epigenomics in my slides, but yes, that is also one of the very important field where you are studying DNA methylation or acetylation changes, and but it is also taking up chip sequencing is mostly for that one only. So, but in companies, a lot of epigenomic projects I have not seen. There are few options in epigenomics in companies, but a lot more in academia. This is what I have seen, but I don't know how it is in many companies. I know a little bit about general projects that happen all across the companies, and I know details about my company. So epigenomics wise, I have not seen many projects in corporate, but there are some. And academia do a lot of epigenomics also.
Okay. So then there is another question from a participant who is a PhD in computational, who is doing a PhD in computational chemistry, and he's asking whether it is possible for, say, a PhD student do a collaborative project with MEDGENOME, or maybe like personally with one of you. Like, so is it possible to do anything like that?
It is very much possible, and Metronome also does the service providing part as a company. So we do that on a regular basis. There is a different team for that, which is called a commercial team, that they collaborate with the academic institutions and help them analyze their data, or just generate the data and give them as per their need. So this is a regular thing, and there are other companies also doing this. So this is possible. Individually, from an employee, it may not be possible because the employee also represents the company, and we are legally bound to not to do thing in which the company is working on. But with the company, it is very much possible, and it is happening on a regular basis.
And there is next question. Is it possible to do PhD through pure bioinformatics work without any wet lab work and only with the data generated through a certain project?
It is possible, but then the thing is, you have to convince your examiner and convince the people you're defending your viva to that your analysis, bioinformatics analysis, are just predictions. They are not benchmarked, validated interpretations of the data. They are predictions of the data, that this data is likely to be something. This phenotype is likely because of this particular genotype. So this is what bioinformatics tells. So unless you validate it through wet lab, it is not benchmarked. So people will not be able to trust that result unless it is validated. So if you want to avoid wet lab experiments in PhD for certain reasons, then you will have to use multiple different bioinformatic approaches to validate your results. That will be a must. And there are also, you can also utilize assay results from any other lab which are published, and then correlate your, I mean, which is related to your data set. For example, if you didn't have, say you're looking at certain SNPs. So people have done a lot of wet lab assays and published SNPs and deposited them online. So you can utilize those and then say that, we also found at this position the same thing, so it is validated. So if these few SNPs are validated, the other also which got predicted are likely to be high confidence calls. So you have to correlate your data with directly or indirectly with those, either directly with some assay data, experimental data, or indirectly you have to validate it through multiple different approaches in bioinformatics. But some concordances should be there. You cannot just stick to one method and say that this is a useful analysis. It will be difficult to accept that.
Yeah. So maybe I would add from my own personal experience, because I did my PhD with almost entirely data-based work where I didn't do any wet lab myself, but we had collaborators who generated some data and then we analyzed it. So those
Yeah.
sort of things can also happen, and there are also PhDs which can be completely focused on data analysis. But as Dr. Meeta mentioned, usually you are expected to look at it in different angles with different kind of analysis to prove the point that you are working on. And at the same time, there are certain analysis, for example, say phylogenetic reconstruction, which probably you can never do with a wet lab analysis, right? Unless you are doing, say, like
SNPs and
experimental evolution. So there are different kinds of projects one can do as a PhD student in bioinformatics. But yes, it depends on the lab where you do that work and how you take it ahead with your PI. So that is something to keep in mind, that genomics has both wet lab as well as dry lab aspects to it. And the most successful projects that I have seen usually involve working with both of them. But yes, you can do either of the two also if you have collaborators working on the other aspect. So that's just one
and
point from my side. Yeah.
just with your points, Nehal, I also remembered one more point. It is
Mhm.
completely possible to do your PhD in dry lab only if your PhD is also oriented towards a tool development or a method
Yes.
development. Then you don't need any wet lab. Although there also, if you're able to do wet lab validation, it will be great, but it's not uh,
Yes.
mandatory.
Yes.
So it also
That's
depends on what you're working on.
Yes, and with so much data now publicly available, I think there is a lot of scope for machine learning, deep learning based like data science projects as well as tool development projects. So yes, a lot can be done, and I think you should just keep your eyes open and explore, do internships, and I think it will be a nice experience for you to work with genomics. Um, yeah, so I think we have covered almost all the questions till now. And yeah, we have covered all the questions on the chat session as well. So it was really a great talk with you here, and Dr. Meeta has covered all the important aspects about working in the area of genomics, taking from what kind of skills one needs to thrive in genomics, say uh, having the adaptability to learn newer techniques, computational skills, as well as wet lab techniques, and also what kind of applications genomic has, and what companies in India are doing right now. So I hope that all participants had a nice overview of how genomics is growing in India, and if they would like to do their career in it, I think it is a great time right now. And so, from on the behalf of LLB School, I would really like to thank Dr. Meeta for accepting this offer to give the talk on genomics, and giving a really nice, comprehensive talk on it. So, thank you, Dr. Meeta. It was a really
Thank you.
pleasure.
Thank you. My pleasure. My pleasure.
Yeah. So I think that's it from LLB School. We have already shared the quiz link with you all here in the chat session as well as on our Telegram channel. So please take the quiz, and all those who score more than 50% would get a certificate. So thank you all for joining here. Bye.
Bye. Thank you.